Hey everyone, welcome to your exam 1 review for your biochemistry lecture 3050. I am your lecture TA. My name is Cassandra, for those of you who have not seen me yet. Um, I am a second year PhD student in the biochemistry and genetics program, and I work on computational genomics, so lots of data analysis, and currently working on the Y chromosome. In my free time, I like to do a lot of outdoorsy stuff. I like to snowboard, hike, mountain bike. I like to go on walks with my puppy Duke. He's 1 year old right now, so still a little pup. And last year and the years before that, I was a track and field athlete. I competed for Clemson in long jump, but sadly I am out of eligibility now. Yeah, if you have any questions, feel free to come to my office hours. Tuesdays, I hold them on the second floor of LSF in person, and Wednesdays and Thursdays, I hold them over Zoom. If you have any questions outside of that, feel free to email me whenever, and we can also set up a time to meet. All right, so let's jump right in. What is biochemistry? Basic enough, it is a mixture of biology and chemistry, and it focuses on the structure and function of molecules. So it's important that you guys understand the basics of chemistry and biology. This includes knowing how reactions work, that they are driven by a change in free energy, as well as enthalpy, and that many reactions in living systems are coupled to make them energetically favorable. Often catalysts are necessary to drive reactions to completion, and in living systems, enzymes often act as catalysts. Also know that there are different categories of cells, that there are prokaryotes and eukaryotes, and understand the basic key differences. Know that living organisms are comprised of macromolecules, and that biochemistry is about acquiring the transformation, usage, and storage of energy, as well as passing information on over generations in the form of DNA, for example. Important elements and biopolymers in living systems are carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, and they make up most compounds found in all organisms. It's important that you know which functional groups are commonly found in biopolymers. That would include aldehydes, OH groups, carboxyl groups, that kind of stuff. And also know that there are 4 major types of biological molecules, which would be amino acids, carbohydrates, nucleotides, and lipids. So our first group are amino acids. I'm sure you have seen this multiple times before. Know that they have a carboxy group, which is on top, and a amide group, which is at the bottom, and that they are amphiphilic molecules, which means they can carry positive and a negative charge. And know that there is a R group, which in this case is alanine, is just a CH3 group, but is oftentimes a little bit more complex, can carry different charges, and the R group determines the properties of the amino acids. Okay, and there are carbohydrates, which are chains that are made up of more than 3 carbons. They have a carbon, a hydrogen, and oxygen. So it's those OH groups and H groups right here. And they can be drawn as Fisher projections, which is just this basic model right here, or they can be drawn as cyclical Haworth projections. Just know that if you see these represent the same molecule, just in different projections. Our next form would be nucleotides, which are made up of carbon, nitrogen, phosphorus, hydrogen, and oxygen. They have a nitrogenous base, a 5-carbon sugar, which can be deoxyribose or ribose right here. And then they have a phosphate group attached to it, which can be anywhere from 1 to 3 phosphates. And next we have lipids, which can have a very wide variety of structures. They're usually just very, very long hydrocarbon chains. A super important one for us, for example, are sterols. So just know how the basic structures of those molecules look. Okay, so what are polymers and why do we make them? Why do we have them? So polymers are made by forming covalent bonds between monomer units that then make up long chains of molecules. In living organisms, there's 3 kinds of polymers. First, we have amino acids that can be linked into polypeptides and proteins through peptide bonds. Then we have nucleotides linked into nucleic acids via phosphodiester bonds. And then we have monosaccharides, which can be linked into polysaccharides or carbohydrate sugars, which can be linked through glycosidic bonds. Lipids, however, do not form polymers. This is super important. So just know that only amino acids, nucleotides, and monosaccharides can form polymers. Lipids cannot. Also understand, why do we make polymers? So essentially, we can make an infinite amount or diversity of structures formed from a limited amount of starting material. So we can also recycle those units. We can break them down into their monomers, and then we can make different kinds of structures from those same units without having a million different types of monomers. So that's super important and allows for lots of unique properties. Another super important basic are hydrogen bonds. Just to know that water has tetrahedral geometry, which makes it polar. So oxygen is partially negative, hydrogen is partially positive charged, and neighboring water molecules will align themselves in a way that they have their opposing charges facing each other. And then those hydrogen bonds can form between the negatively charged oxygen and the positively charged hydrogen. And also know that you don't necessarily need just oxygen or hydrogen to form those bonds. This also works with oxygen, nitrogen, and sulfur, because those are also partially negatively charged molecules. It is, however, important which other atom those are bound to in order for them to be partially negatively charged, or the hydrogen to be partially positively charged. So if a hydrogen is just bound to hydrogen, there's no partial positive charge. They are only partially charged if they are bound to a more electronegative atom. Okay, and then other different kind of bonds are London dispersion forces, which can form between non-polar molecules. They result from the formation of temporary dipoles and the movement of electrons, which would be induced dipoles. Van der Waals forces, London dispersion forces are a type of Van der Waals force. So essentially it's the same thing. Weak forces between molecules due to temporary or permanent dipoles. In general, longer molecules such as super long carbon chains can form really strong non-polar attractions. And as the chains grow longer, the forces can actually get pretty strong. Then we have hydrogen bonds, which we just talked about, ionic interactions, which would be between positively and negatively charged molecules, and then an actual covalent bond in which there's an actual bond happening. Okay, so then we're going to jump right into nucleotides. Know that there are 2 different types. There is purines and pyrimidines. One of them is a 1-membered ring, the other one is a 2-membered ring. Adenine and guanine are purines, so they are 2-membered rings. Cytosine, thymine, and uracil are pyrimidines. They only have a single ring. Know that DNA contains those 4 nucleotides, so A, C, T, G, and that RNA contains uracil instead of thymine. And then also DNA and RNA contain pentose sugars, which I already mentioned earlier. So DNA has deoxyribose, hence the D, and RNA is a ribose sugar. So again, nucleotides have a sugar backbone, so either a ribose or deoxyribose. The base is attached at the number 1 carbon. So right here, this is the sugar ring. This is the number 1 carbon with the base attached to it. So this can be adenine, guanine, cytosine, thymine, or uracil, depending on if we're having DNA or RNA. And then the phosphate is attached at the 5' carbon, which would be right here. And then on the number 2 carbon right here, this is where we would determine if it's a DNA or RNA. So if it's deoxyribose and it's missing a hydrogen, or if it's OH in ribose. And then the chain gets elongated on the 3' carbon. So if we link nucleotides together via phosphodiester bonds, this is exactly how that happens. So phosphate on the 5' carbon is linked to an OH group on the 3' carbon, which is where we always link from the 5' to the 3' direction. And the open end of the chain always has that free 3' OH. So here is where that next phosphate group would then form a phosphodiester bond. DNA is super important, that we know that A pairs with T and C pairs with G. So that is a very basic and super important rule. And therefore, the A plus G content and C plus G content are always the same. So purines, the amount of purines in DNA equals the amount of pyrimidines. And the structure of DNA is, there are 7 basic facts. These strands run anti-parallel to each other. So 5' to 3' and 3' to 5'. It is a right-handed helix, so it is counterclockwise, and has a very consistent diameter of 20 Angstroms, and a full turn of a helix is 35 Angstroms or up to 10 base pairs. There's a major and a minor groove, and the DNA binding proteins can then bind to that major groove right here, obviously. The sugar phosphate makes up the backbone, so this right here is a sugar phosphate backbone, and the bases stack perpendicular to the backbone in the center of the helix. Also know that the backbone has a negative charge thanks to the phosphate, and that magnesium ions can bind near that phosphate. Structure of RNA is that it is single-stranded. Know the most important differences between DNR and RNA, different base pairs, single versus double-stranded. RNA can form secondary structures, and there are different temperatures. So those secondary structures, for example, right here, it can fold into itself and form temporary double-stranded bonds and different kinds of structures. Also know that DNA and RNA can form a hybrid helix, so they can form double strands with each other. So base pairs can form between A and U and C and G. And DNA stability depends on those stacking interactions. So like I said, the bases stack in the middle, and those stacking interactions lead to the stability of DNA. Under certain temperatures, DNA can unfold or denature. So that temperature is called TM, temperature 2, that was required to disrupt 50% of the structure. So in this case, that will be right here. So 50% of DNA would be denatured. And the higher the GC content, the higher the melting temperature, because GCs form stronger base pairs because they have 3 hydrogen bonds versus A and T. So therefore they need more energy to be disrupted. Under cooling temperatures, DNA can renature, so it can form a double strand again. However, it's important that it is cooled slowly, because if it is cooled too fast, improper base pairing can be the result, which then we have to heat it again, denature it again, and then cool it slow for it to be in the correct structure. All right, moving on to the next important topic, genes to proteins. So the very basic concept of biochemistry in all living things is that DNA is transcribed into RNA, which is a transient molecule, which is then translated into a functional unit. Sorry, this is supposed to say functional protein unit. I don't know why that is in the middle of this word in here. So yeah. So the genetic code is 3 base pairs encoding 1 amino acid. And you see why it is super important here to know that mutations in the genetic code, so in DNA, can cause diseases that actually have a pretty big effect. So like I said, DNA is translated and then transcribed and then translated into a protein. So if there are any errors in the DNA sequence, that directly affects the protein that is being made, which then affects or can have phenotypical effects. Doesn't have to though. But an incorrectly paired base can very quickly lead to a mutated protein. Also, it's important to know that only about 2% of the human genome is actually encoding for proteins, and over the majority of the genome is made up of very unique sequence or very repetitive sequence. So only 2% is actually transcribed into a functional protein. And then there are different forms of DNA manipulation that can be done. For example, we can use restriction enzymes. Those recognize restriction sites and can create sticky ends. We can visualize DNA on gel images using gel electrophoresis. We can clone and produce recombinant DNA using restriction enzymes and vectors. We can amplify DNA via PCR, and we can sequence DNA, for example, via shotgun sequencing, Sanger sequencing, or next generation sequencing. All right, so we're going to talk about DNA replication and repair for a second. And in general, what is a gene? It's a physical and functional unit of heredity and often includes regulatory sequences. So that is encoded by DNA, and in order to have enough DNA in every cell, we need to make copies of it. We need to prepare the cell for cell division, and based on the structure of DNA, this can be accomplished by or thanks to the double helical structure. So there's 2 different templates, and thanks to the complementary base pairing. So if we take one strand, because it can form complementary base pairs, it is pretty easy to create the complementary strand from that template. And what we need for that is obviously our parent DNA strand. We need DNA polymerase. We need deoxynucleotides. So DATP, TTP, DCTP, and DGTP. And DNA is replicated via the semi-conservative mechanism. So each strand serves as a template for the new strand. So we have the parental DNA, and then each of those strands can serve as a new strand. So the parental DNA is denatured, and then we make a strand of each of the parental strands. Replication always happens in the 3' to 5' direction. And also important is that the replication machinery is stationary and that the DNA moves along the replicase. So the DNA is pulled along the DNA polymerase. Okay, so how do we start replication? In prokaryotes, that is fairly simple because DNA is organized in circles and there's only one origin of replication. So right here, and then we form 2 replication forks and essentially the replication forks move in both directions, and then we get 2 new circles of DNA. In eukaryotes, it's a little bit more complicated because DNA is linear, so we have many different origins of replication wherever it tells us to start, and we have many replication forks, and then we get many of these replication bubbles right here. So these are different origins of replication, and then we have different replication forks forming. Okay, so which proteins are needed for replication? Oh, sorry. We need helicase, which is ATP dependent and can unwind DNA. Also, it disrupts the H bonds. Like I said, we have those double-stranded strands, and we need to unfold them in order to access each template. So because ATP is required here, that process is irreversible because we are spending ATP on unwinding the DNA. And then since we are unwinding it, we kind of have to imagine that there's a lot of tension that's being built up since the DNA is kind of wound around each other. So we need a topoisomerase that relieves the strain caused by the unwinding of DNA. And then we also have single strand binding proteins in prokaryotes, which is important because otherwise those single strands are very susceptible to attack by nucleases, or they would just reform base pairs. So we need proteins that bind to those single strands in order to keep them apart. In eukaryotes, that is done by the replication protein A. It's essentially the same thing as a single strand binding protein, protects single stranded DNA from nucleases and prevents secondary structure formation. So prokaryotes have 5 different DNA polymerases. Only polymerase 1 and polymerase 3 are really important in this process. Eukaryotes have multiple different DNA polymerases and are denoted by Greek letters. So alpha, epsilon, and delta are super important for replication. DNA can only extend a pre-existing chain. So we need a primase to make primers. Those primers are made up of RNA, and then DNA polymerase can extend the chain from those primers. So a 20 to 30 nucleotide RNA fragment is made by primase, and then that chain is extended by DNA polymerase. So it cannot initiate polynucleotide synthesis, but it needs that primer. It can also only work in the 5' to 3' direction. So there are going to be 2 different strands, a leading and a lagging strand, because it always requires that free hydroxyl group of the phosphate for the phosphate to attach the next nucleotide. So we talked about that earlier, how the phosphate on the 5' carbon attaches to the free hydroxyl group on the 3' carbon. So like I said, we have a leading strand right here, which would be continuous. So the primer is made right here, and then DNA polymerase can extend the strand in pretty much one go. In eukaryotes, that is done by DNA polymerase epsilon. In prokaryotes, that is done by DNA polymerase 3. And the lagging strand right here produces those little fragments, which are called Okazaki fragments. They are discontinuous, as you can see. So because it can only work in 5' to 3' direction, so it can only extend from 5' to 3', but that strand obviously is opening up in the other direction. It has to make, so primase makes primers, and then DNA polymerase reads in 5' to 3' direction, makes a little piece, and then a new piece of strand opens up, so then it has to jump back and makes another Okazaki fragment. The primers are then degraded by RNase H or DNA polymerase 1, and DNA polymerase 1 replaces them with DNA. In prokaryotes, polymerase 1 has exonuclease activity, so it doesn't need that RNase H that is needed in eukaryotes. RNA ligase then connects the DNA fragments, so those Okazaki fragments right here, and makes them into one continuous piece. It is also important that there is proofreading, because we can accidentally incorporate the wrong nucleotide. So there is an error rate of 10 to the 10th. So it is important that DNA polymerase has a proofreading ability, which it does, and it can excise in the 3' to 5' direction. So it can excise misplaced nucleotides and polymerase will then go back and replace them. So here's the entire process, an overview. So we have helicase, which unwinds in this direction right here. Topoisomerase relieves the strain that is created. Then we have single strand binding proteins binding to the single strands and making sure that they stay single stranded. And then on our leading strand, we have a single DNA polymerase 3 that slides along, well, pulls the DNA along. It doesn't slide, it's stationary. It pulls it along. And we have that sliding clamp that makes sure that the DNA polymerase stays in place and doesn't fall off. And then on the lagging strand, we have multiple fragments that are being made. So we have primase that makes a primer, and then DNA polymerase makes a small piece. And then we can see that there are already pieces of DNA that were made. DNA ligase is connecting those pieces, and DNA polymerase 1 is replacing the primers and making DNA instead of RNA. Understand the differences between prokaryotes and eukaryotes. So know that there are different proteins, and know that prokaryotes have circular DNA, so it looks a little bit different than eukaryotes. Okay, next step would be transcription and RNA. So, like I said, the central dogma is we have DNA and it is transcribed into RNA, which is a transient molecule and serves as like a sort of template to then make protein. So in eukaryotes, we have a nucleus. So we have replication and transcription happening in the nucleus. Translation then happens in the cytosol. So they are spatially separated and happen sequentially. In prokaryotes, we don't have a nucleus, so it all happens simultaneously. Replication and transcription happen in cytosol, and so does translation. So as soon as DNA is being transcribed, we immediately have ribosomes essentially attaching to that transcript and making protein as it goes along. Okay, so then there are different types of RNA. We have mRNA, which translates into protein. And then we have transfer RNA, which carries amino acids and then translates RNA into proteins. We have ribosomal RNA, which is a part of the ribosome, as the name suggests. And then we have lots of non-coding RNA, which can be important for the regulation of gene expression in different ways. Also know that DNA is very, very, very highly compacted because it can be up to 2 meters long if it is stretched out in a single piece. And we need to somehow fit that into our tiny little cells. So they are wrapped around nucleosomes and supercoiled many times to compact them really, really small. However, we need to make them accessible for transcription. So we need to make that DNA, we need to package it looser, which would then be euchromatin. Euchromatin is transcriptionally active. It is loose, and the nucleosomes are moved to make it accessible for proteins that are being transcribed. And then we also have heterochromatin that is very, very dense still, and there is no transcription happening there. All right, a quick overview of what a gene is. Like I said earlier, it is a functional unit, and it includes information about the sequence of an amino acid. So part of that is we have a promoter region, we have that RNA coding region, we have a terminator right here, and we have upstream and downstream sites of the gene. Again, mRNA is transient and can be degraded very, very quickly in the cell, and therefore that is also a way that gene expression can be regulated by the rate that RNA is degraded in a cell. Okay, so what is required for transcription? We need transcription factors, we need activators and co-activators, we need repressors, we need enhancers and insulator sequences, we need mediators, we need RNA polymerase, we need nucleotides, the DNA template, obviously, and we also need a termination site so we know when transcription is over. Okay, how do we initiate transcription? We have, like I said, a promoter sequence, which is a DNA sequence that is recognized by the transcription machinery. Oftentimes in prokaryotes, that is that Tata sequence. I'm sure you've heard the name Tata box before. So that is that region right here, which is usually up to 10 base pairs upstream of the transcription start site. And then there's another transcription sequence that is super important, that is 35 base pairs upstream, which is the TTGACA region. But the Tata box is usually a lot more conserved in this region, that -35 region right here. There are also lots of other sequences that can be super important for transcription, that BRE sequence, that initiator sequence that is usually found downstream of the transcription start site, and other elements that can also be found downstream, so within the gene actually. Okay, so then the transcription machinery seems quite complicated, but actually really isn't. So RNA polymerase is assembled from multiple subunits. It has 2 beta units right here and 2 alpha units right here. And that enzyme right here, so those 2 beta and 2 alpha units are called the coenzyme. And then we have that sigma factor, which is important for the promoter recognition. There's different kind of sigma factors, and they can recognize different kind of promoters depending on the gene. So there's different ones, for example, that can like recognize just household genes that need to be on all the time, or like in case of emergency, there's certain genes that need to be turned on, and there's different sigma factors. So when that sigma factor is bound to the coenzyme, it forms that holoenzyme. Okay, and the way this all works is that we have that Tata box and that transcription factor 2D. So these general transcription factors are all named TF2, and then they have different letters. So transcription factor 2D binds to that Tata box, recognizes it. There's also another protein which is called Tata binding protein, TBP, sorry, and they bind to that Tata box. Then we have TF2A, which then binds to TF2D. TF2B then recruits the pre-initiation complex, which is the RNA polymerase 2, and TF2H and TF2F. And TF2H has helicase activity, hence the H, but I'm pretty sure you guys don't need to know what all of these units do. Just know that they exist and that they help in recruiting the pre-initiation complex to the promoter. And so then at the end of all this, we have that assembled pre-initiation complex with RNA polymerase at the transcription start site. Okay, so how do we regulate gene expression? Because obviously we don't need all genes turned on at all times. That would just be a waste of energy, especially in bacteria. We need to be able to conserve our energy. So we have in eukaryotes enhancers and silencer sequences, which usually sit upstream of the gene. And then we have activators or repressors that can bind to those sequences, and they aid in increasing or decreasing transcription by, for example, recruiting other proteins that can aid in transcription, or by blocking RNA polymerase from binding, or from forming a secondary structure. So there's multiple different ways that that can happen. And then we have a mediator that connects the enhancer and silencer with the pre-initiation complex. So right here, DNA bends the activator that is bound right here to the enhancer sequence, then interacts with the pre-initiation complex right here through that mediator. So important regulatory sequences. Like I said, especially in prokaryotes, gene regulation is super important because we don't need to waste energy when we don't have certain substrates present. So, for example, one of those ways is the lack operon. So usually we have operon control, which means that more than one gene is controlled by an entire promoter complex. So, for example, here, 3 different genes, the LAC-C, LAC-Y, and LAC-A, which are needed for the metabolism of lactose, are all controlled by the same operon. So when lactose is absent, the LAC repressor binds to the operator, and RNA polymerase cannot bind because that repressor is in the way. However, when lactose is present, a little complex called allolactose binds the repressor, and then that allosterically modifies the repressor so it cannot bind to the operator anymore. And then RNA polymerase is free to bind to the operator, and it can transcribe the genes that are needed for the metabolism of lactose. Like I said, this is super important because if no lactose is present, why would we want to transcribe these genes if we don't need to? Okay. The same way there are different DNA polymerases, there are also different RNA polymerases. In eukaryotes, different types of RNA polymerases make different types of RNA. I've mentioned earlier that there's ribosomal RNA, which is made by RNA polymerase 1. mRNA is made by RNA polymerase 2, and then tRNA and other small RNAs are made by RNA polymerase 3. RNA polymerase 2, the one that makes mRNA, has a clamp protein, and that allows for very high processivity. So that just means it can process RNA pretty quickly without just falling off. And it also has backtracking activity, which means it can proofread and correct mismatched errors. So like the DNA polymerase, it has a 3' to 5' exonuclease activity. Okay, so then how do we go from transition to elongation? From initiation to elongation, sorry. So that transcription machinery, that pre-initiation complex that we had, is very firmly associated with the promoter, thanks to that sigma factor also. So we need to release that sigma factor. So DNA is scrunched multiple times, so that pre-initiation complex tries to slide RNA along, that DNA along to make RNA, and so that DNA is scrunched, and eventually the sigma factor is released, and that enables the transcription machinery to move forwards. And that transcription factor 2B that we had earlier must move for the active site of RNA polymerase to be available. So it needs to rearrange a little bit. And then the C-terminal end of the RNA polymerase gets phosphorylated, which requires ATP and makes that step irreversible again because we are spending energy to make that happen. So once that C-terminal is phosphorylated, we can start elongation. It's also important to know that the coding strand of DNA, the 5' to 3' strand, is identical to the RNA transcript. It's the only difference is that U is replaced for T, but besides that, the sequence is entirely identical. Okay, and then we also need to terminate transcription, otherwise we would just make a ton of RNA that we don't need, and we would not be able to make the proteins that we want to make. So there's 2 different types of termination. One is row dependent, which is just a factor called row, and it slides along the mRNA, so it binds here, and it slides along the mRNA until it reaches RNA polymerase and just pretty much knocks it off. And then there's one that is row independent. There is a GC-rich sequence that destabilizes the DNA RNA helix, and the mRNA dissociates as that RNA polymerase pauses. The DNA and mRNA dissociate, and essentially that destabilizes the helix, and mRNA is cut off, and RNA polymerase is essentially also knocked off. Okay, and in eukaryotes, it looks a little bit different. RNA polymerase slows down once it sees a poly-A signal. So that poly-A signal, we'll get to that in a second, and I'm sure you guys remember this from class, is needed to make that poly-A tail that we need for RNA processing. So once RNA polymerase encounters that signal, it pauses, and during that pause, proteins bind to the phosphorylated C terminal and they cause termination. So mRNA dissociates and then exonuclease starts degrading mRNA until it catches up with the polymerase and then it pushes it off the template. Okay, and then, like I said, we need to process RNA in order to go from the pre-mRNA to mature mRNA. So we need to add a 5' cap, which is a methyl G added to the 5' end, and it protects the 5' end from degradation by exonucleases. So this essentially happens right as that 5' end is being made. There's a little signal in the upstream sequence, and there's a 5' cap being added to stop it from degradation. And then we have a poly-A tail, which is 60 to 200 adenines long. So poly-A stands for polyadenine. It adds stability to the transcript and can control the time of degradation. So that is done by the poly-A binding protein, PABP. And then we add that poly-A tail. And so then our mature RNA has that 5' cap and that poly-A tail. And then to degrade RNA, we have a decapping enzyme, which removes the 5' cap. Then we have deadenase, which removes the poly-A tail. And then we have exonucleases, which aid in the decay from either side. It just starts chopping down mRNA. So then that mRNA is degraded. And then we have splicing, which allows for different proteins being made from the same transcript, essentially. So we know that there are introns and exons, and we only need exons that actually code for a protein. So introns are pretty much the sequence that is not actually being translated and that we don't actually need. It is, however, transcribed in the process. So then we have a spliceosome, which can cut out on the splice sites right here. And then it connects only the exons together. So that intron is cut out and then exon 1, exon 2 are connected. And this is super important because, like I said, we can make multiple different mature RNAs from the same transcript. So we can have very different types of proteins. Well, not types of proteins, but very different proteins being made from the same transcript. Okay, and then the final step in this whole process is translation. We have nucleotides that need to be translated into protein, so amino acid sequence. And I have mentioned tRNA before, which acts as a translator. So we need to know how the genetic code works. And you guys should have seen that table where we have different 3 base pairs that encode 1 amino acid. And it is also important to know that the code is redundant, so that each amino acid is encoded by more than 1 codon. A codon are those 3 nucleotides, and that it is universal, so it's the same across all species. And it is non-overlapping also. So 1 codon always just codes for 1 amino acid, and then it moves along 3 at a time. So only 3 code for 1 amino acid. So again, the tRNA acts as a translator. So the tRNA has an amino acid bound to it and recognizes the correct codon. I will get to that in a second. And again, it's important to know that mRNA is arranged 5' to 3' so that the tRNA can recognize the codon properly, and that we have 20 different amino acids, and those allow for 64 codon possibilities, thanks to that 3-to-1 translation. Okay, again, tRNA carries amino acid. So this is the structure of tRNA, so it is a form of RNA, and it forms those secondary structures that I've mentioned earlier. So it can fold in on itself, and on the 3' end, it has a free hydroxyl group where an amino acid can be attached, and then it has that anticodon right here, which is complementary to the codon sequence, so the sequence on the mRNA, and that corresponds to the correct sequence and can therefore bring the correct amino acid that is needed in the sequence. The way tRNA gets amino acylated, it's a synthase reaction. So the amino acid reacts with ATP to form an amino acyl adenylate via hydrolysis of phosphate. And this is again an irreversible step because we are spending energy. And then that activated amino acid reacts with the tRNA to form an amino acyl tRNA and adenosine monophosphate, so AMP. So again, we spend energy to make this happen, and therefore it is an activated tRNA or amino acylated tRNA. So we covalently add an amino acid to the acceptor stem of that tRNA, and it is done via an ester bond. So that's also important to know. And amino acyl tRNA is the protein or enzyme that carries out that reaction. Also important to know is the wobble hypothesis. So that means that most tRNAs can bind multiple codons because only the first two bases must be a specific fit. So only the first two bases actually need to fit perfectly. So the first two bases of the codon need to correspond to the anticodon, and the last one can actually be a mismatch. So that allows for different amino acids to still be added to the same codon. And this is super important because mutations in that last amino acid don't necessarily lead to the wrong protein or the wrong amino acid to be integrated. So it can lead to silent mutations that actually have no consequences because the correct amino acid will still be integrated into the sequence where it should be. And then we need a ribosome to actually make that process happen, like the process of translation. So I've mentioned before, we have rRNA, which is ribosomal RNA, which is obviously needed for the ribosome. So the ribosome is made up of RNA and protein, but it has a higher percentage of RNA. And eukaryotes and prokaryotes have small, well, the ribosome has small and large subunits. There are different subunits in each type of cell. So prokaryotes have different subunits than eukaryotes, and they have a different sedimentation than okay, and then the last important thing is how do we assemble that ribosome, and how do we actually start the process of translation. So there are 3 TNR binding sites in the small subunit of the ribosome. There is an A site. This is where the charged tRNA will bind initially. Then we have the P site, which is where the peptide bond is formed between the growing chain and the new amino acid. And then we have the E site, which is where the empty tRNA exits. I feel like this is pretty easy to memorize because P is where peptide bond forms and E is where empty tRNA exits. So then A is obviously where the acceptor site is. Okay, and so here's how that entire process comes together. So we have the initiation of translation where the small subunit scans the mRNA for the start codon, which is always methionine. So that AUG right here, codes for the methionine amino acid. And once that is found, the tRNA carrying that initial amino acid will bind to the P side, not the A side. The P side in this case. Oops, so sorry. And once that is bound to the P side, the large subunit follows, and the ribosome is fully assembled. Sorry if you hear my dog snoring. And then elongation continues, so the next charged tRNA binds in the A side, and then a peptide bond is formed between that growing chain in the P side and the new incoming amino acid. It comes into the A side, and then the ribosome moves 3 nucleotides along, and then the empty amino acid will sit in the E side, the growing chain in the P side, and the E side will be open. And then that entire process also needs to be terminated because, again, we don't need to move along the entire strip. So there's a termination sequence, which happens when the final polypeptide is fully synthesized. And the chain, the entire polypeptide chain, is cleaved off of the tRNA that carries the entire polypeptide chain. And once that happens, the ribosome is disassembled. Okay, I think this is it for our review. I hope I covered most of what we went over in the lectures. I tried to do my best to keep this pretty concise, and I know that I probably didn't hit every single topic, but I do hope this helps. And if you have any more questions, feel free to join office hours or to send me an email and we can set up a meeting. So yeah, good luck on your first exam.